Hello students, today we start from that is our molecular base of genetic intelligence, this chapter. Now we are going to start from machinery and the enzymes. Machinery and the enzyme. Which machinery? It is DNA replication. How a DNA replicate itself, how a new DNA is formed, and which enzymes are responsible for this process. Okay. And one thing you know very well, DNA replicate. When does DNA replicate? First question, when does a DNA replicate? The answer is very simple, when a cell divide. At that time, cell divide means division means any type of division. It is a mitosis or meiosis, right? DNA divide. DNA divide means DNA replicate. We cannot say it is divide, but we have to say that DNA replicates or duplicates itself as the phase S phase, right? This is the S phase when DNA duplicates. At that time, the number of chromatin threads, it becomes 4n in a diploid cell, right? Meiosis means 4 gametes are formed. That means they are haploid. Mitosis means they have equal number of chromosomes. We know very well. Number of chromosomes is not increased, but number of chromatin threads, that means number of DNA content is double. Double means what? It duplicates itself. It forms a new copy of itself, right? And in the previous line, we understand that a DNA replication is a semi-conservative process. Means one parent strand is there and another it is daughter strand, right? In DNA replication, one thing is there. Here, both strand act as template strand. Okay. Now come to here in NCERT, machinery and the enzyme. How a DNA replicate and which enzymes are responsible for this replication, right? In living cells, such as E. coli. E. coli means what? Prokaryotic cell. The process of replication requires a set of catalysts. In biology, we said them enzymes. The main enzyme is referred to as DNA-dependent DNA polymerase. DNA-dependent DNA polymerase. This process is totally dependent upon DNA, right? So it is called a DNA dependent because for this process, at least we need one strand of parent DNA, right? And DNA polymerase, because here DNA polymer is formed. So this enzyme is called DNA polymerase. Already we know, I tell you here that ASE is the suffix which is used for enzymes, right? You understand here? DNA dependent DNA polymerase enzyme is responsible for DNA replication. Since it uses a DNA template to catalyze the polymerization of deoxynucleotides. Here, because DNA replication occurs, here it needs deoxynucleotides, right? That means what? We have 4 nucleotides which are deoxy, that is deoxy adenosine triphosphate, deoxy guanosine triphosphate, deoxy thymidine triphosphate, and deoxy cytidine triphosphate. These are 4 nucleotides which are essential for DNA polymerization, right? These enzymes are highly efficient enzymes as they have to catalyze polymerization of a large number of nucleotides in a very short time. Very fast, very rapid reaction is there. E. coli that has only 4.6 into 10 power 6 base pairs, right? When it compared with human whose diploid content is 6.6 into 10 power 9 base pair, completes the process of replication within 18 minutes only. That E. coli, it complete the replication means a DNA convert into 2 DNA, a single DNA convert into 2 DNA within 18 minutes, right? That means the average rate of polymerization. Here you can understand how rapid this process is there, right? This process, how rapid this process is. This E. coli that has only 4.6 to 10 power 6 base pair, when it is compared with the human whose diploid content is 6.6 into 10 power 9 base pair, it completes the process of replication within 18 minutes because 20 minutes means E. coli get double. 2 cells are formed, right? That means the average rate of polymerization, formation of extra or another DNA, to be approximately 2000 base pair per second. How rapid? We cannot imagine. 2000 base pair per second. And another thing is there, accuracy is also. Not only do these polymerases have to be fast, but they also have to catalyze the reaction with high degree of accuracy, very high degree of accuracy. No mistake occurs here. Mistake means what? Mutation happens, right? So no mistake is there. Per second, 2000 base pairs are formed, right? So you can understand here, if you want to, what is the length of DNA, right? Per second they forms. So you can multiply this 2000 base pair, multiply by 3.4 angstrom, and then you can get the length of DNA which is prepared in 1 second. Okay. Any mistake during the replication would result in mutation. Already I tell you. If a minute mistake means, if a single nucleotide mistake, mistake means what? A single nucleotide mutation happens here. Like sickle cell anemia, only a single nucleotide mistake is there, right? Major mistake means it becomes a suppose that one slip off, that what a frame shift mutation occurs, right? Point mutation, frame shift mutation, a very single nucleotide, that is, if it is replaced by any other nucleotide, then point mutation occurs. If it is slip off, then what happens? The whole means frame shift mutation occurs, and you know very well in the previous chapter, frame shift mutation means what? The segment from where mutation occurs, the whole remaining segment, it is totally altered, right? That means totally changed. Furthermore, energetically replication is very expensive process. Energy, it requires more energy. Why? Because bonds are formed, and bonds means what? It is energy, right? So, deoxyribonucleoside, look here, deoxyribonucleoside triphosphate, they are deoxyribonucleoside side triphosphate, they are not tritide. When you read, they are triphosphate, right? When you say deoxyribonucleotide, then they are not triphosphate. That means what? They are monophosphates, right? So this is deoxyribonucleoside triphosphate. As for example, adenosine triphosphate, ATP, that is energy molecule. GTP, CTP, TTP or UTP, we can say, right? Serve dual purpose. Two functions are there. Serve dual purpose. In addition to acting as substrate, means it is a substrate. We know these triphosphate, they are substrate. That means what? They are nucleotides, which is responsible, which are add and which are responsible for the formation of DNA. They also provide energy for polymerization reactions. They also provide energy because a huge energy is released, because here only ATP is converted into AMP, not in ADP only. Here two bonds are made. First bond is really means first phosphate group is removed, then second phosphate group is removed, then AMP is formed. AMP or GMP or CMP or TMP or UMP, we can say like this. So these triphosphate, they have dual purpose. First purpose is they act as substrate, and second they provide energy, right? Polymerization. Two terminal phosphates in a deoxynucleoside triphosphates, right, are high energy phosphates. They are high energy phosphates, they release energy, and this energy is consumed in the formation of new DNA strand. Okay, same as in case of ATP. Actually, this is adenosine means what? Adenosine triphosphate, it is similar, it is identical to ATP, right? Adenosine monophosphate, adenosine diphosphate, adenosine triphosphate. Mono means single phosphate group, di means two phosphate group are there, tri means three phosphate groups are there. Do you understand here? What is the function of their these nucleotides? That means nucleoside triphosphates. So they provide energy, they act as substrate. Primary function means they are substrate. Secondly, by breaking these two terminal phosphate group, they provide energy for the formation of new DNA strand. Okay. Now here, in addition to DNA dependent DNA polymerase, what is given here? In addition to DNA dependent DNA polymerase enzyme, right? Enzyme. Many additional enzymes are required to complete the process of replication with high degree of accuracy. Okay, with high degree of accuracy. Other enzymes also respond for long DNA molecule, since the two strands of DNA cannot be separated in its entire length. First one thing is because it is very long, so these two strands they cannot be separated fully, right? They have some specific sites from where DNA can separate, right? And these sites are called origin of replication or initiation site. In prokaryotes, only one site is there, only one origin of replication is there. But in eukaryotes, more than one, we can say many, that is initiation sites are there, or we can say many ori are there. Ori means what? Origin of replication, right? Due to very high energy requirement, because it is a very long strand. So if we want to break this hydrogen bond, all the hydrogen bonds, we need more energy. And when we provide, suppose that we provide this more energy, what happens? This DNA denatures, right? So it is not possible to break the hydrogen bonds of whole DNA, right? So then in long, that means linear DNA means not prokaryotic, it is eukaryotic DNA, which is very long. So many origin of replication or initiation sites are there, from where hydrogen bond breaks and means we can say here replication starts, right? But in prokaryotics, only one initiation site is there, or only one origin of replication is there. The replication occurs within a small opening of DNA helix, referred to as replication fork. This one is called as replication fork. When they break hydrogen bond, what happens? A fork is formed, right? Because this side also hydrogen bond is formed, this side also hydrogen bonds are present, but in between this one, this is a site where hydrogen bonds are break down. Means these two DNA strands they get separate. When they get separate, what happens? Now here, new strand starts. Formation of new strand starts. The DNA dependent DNA polymerase catalyzes polymerization only in one direction. That is it. Always you have to need that this polymerization occurs only in 5'3' direction. That is important. So DNA dependent DNA polymerases catalyze the polymerization only in one direction, and that is 5'3'. This creates some additional complications at the replication fork, right? Consequently, on one strand, the template with the polarity of 3', 5', because we know the both strand of DNA, they are anti-parallel, right? And the DNA dependent DNA polymerase enzyme, it act only in one direction, 5', 3' direction. So then what happens? One complication occurs. In 3', 5', a continuous strand is formed. No problem is there. And this continuous strand, it is called as leading strand. What is called? Leading strand, right? But on the other hand, the template with polarity of 5', 3', right? So there what happens? Here a discontinuous, means small fragments are formed. So discontinuous strand is formed, and this continuous strand is called as lagging strand. This continuous is that is what? Leading strand, leader. This one is lagging strand, right? Because it is formed in small fragments, DNA fragments. These fragments are called Okazaki fragment. What is called? Okazaki fragment, right? And this continuous strand, that means I tell you Okazaki fragment. These Okazaki fragment ultimately they bind with each other, they join with each other to form a continuous strand with the enzyme, and that enzyme is called DNA ligase enzyme. The discontinuously synthesized fragment are later joined by the enzyme DNA ligase. Do you understand here? What is the direction of DNA dependent DNA polymerase enzyme? It is 5', 3', right? Continuous strand is formed on 3', 5' polarity template strand, and discontinuous strand is formed on the strand which polarity is 5', 3', right? Continuously strand is called leading strand. Discontinuous strand is called lagging strand, first thing. And the fragments which are present in discontinuous strands, these DNA fragments are called as Okazaki fragments. They attach with one another, they link with one another through ligase enzyme. That ligase enzyme is called DNA ligase enzyme because here DNA strands are joined together. The DNA polymerase on their own cannot initiate the process of replication. One thing is that DNA is totally dependent on RNA. For replication also, not whole process, but the initiation, right? For starting, they need RNA, specific RNA, and this specific RNA segment is called RNA primer, right? So, this DNA polymerase on their own cannot initiate the process of replication, right? So, why it cannot? Because it needs RNA primer, right? So, first what happens? First, this DNA, it forms RNA primer. RNA, for the formation of RNA primer, it needs RNA polymerase enzyme, right? So, sometimes this RNA polymerase is also called as RNA primase enzyme because it forms RNA primer. So, it is called as RNA primase enzyme, right? So, this RNA primase enzyme, it forms RNA primer. The RNA primer initiates the process. When RNA primer is formed, it separates off from the DNA strand. And when it separates off from the DNA strand, then this DNA dependent DNA polymerase enzyme, it continuously opens the DNA strand and forms the daughter strand, right? Opening of DNA strand is not by this polymerase enzyme, but for the opening of, or we can say break down the hydrogen bond, it is possible due to the presence of helicase enzyme. This enzyme is called helicase, right? Also, the replication does not initiate randomly at any place in DNA. It has a specific site, and these sites are called initiation sites. There is a definite region in E. coli DNA where the replication originates, and such regions are termed as origin of replication. I already tell you, in prokaryotics only one origin of replication is there, but in eukaryotics they have more than one replication. Origin of replication is there. This origin of replication is also called as initiation site. Okay. Because of the requirement of origin of replication, that a piece of DNA, if needed to be propagated during recombinant DNA procedure, right, requires a vector. The vector provides the origin of replication. This vector, it provides the origin of replication, and that vector means we can say RNA primer. Further, not every detail of replication is understood well, but in eukaryotes, the replication of DNA take place at S phase. This one is synthesis phase. We know very well in cell division, in interphase is further divided into G1 phase, S phase, and then G2 phase, and then we have mitotic phase, that is M phase, right? So, G1 means growth first phase, then after which comes synthesis phase. In synthesis phase, we know here DNA replication, second centriole replication, and third replication of histone proteins occurs here, right? Here DNA take place at S phase of the cell in case of eukaryotes, because eukaryotes they have mitosis or meiosis, but prokaryotes they divide through binary fission, and binary fission is not a mitosis, right? The replication of DNA and the cell division cycle should be highly coordinated because it is a genetic. A failure in the cell division, okay, suppose that a cell, it goes into the G1 phase, then after it passes into the S phase. S phase means what? Number of DNA content get double, it becomes 4n, right? But this cell cannot, cell division or cytokinesis never occurs. Then what happens? A failure in cell division after the DNA replication results into polyploidy. That is a chromosomal anomaly, polyploidy, right? Suppose that karyokinesis also, this cell does not undergo into the mitotic phase. Then what happens? A single nucleus, it contains 4n. 4n means tetraploidy is there. That is called as euploidy, right? That is also a mutation, we can say, a type of mutation. Okay. And if karyokinesis happens but cytokinesis not happens, then what happens? Then a polynucleated condition is formed. Polynucleated, as for example in tapetum, more than one nucleus is present, right? Tapetum as we read in our plant. Okay. You will learn the detailed nature of origin and the process occurring at this site. Okay, in higher classes we will read this one. Also, some things we read in our another chapter, right? In biotechnology. Okay, so do you understand here how a DNA replicate is? Figure is given here. One strand, it is both are antipolar, 5' 3' and 3' 5'. In 3' 5' this side, right? So 3' 5' what happens? This DNA polymerase, DNA dependent DNA polymerase enzymes, it forms a continuous process. It get open and this new strand is continuously formed. It open and it forms, right? But what happens here? When a small fragment open, then a strand is formed. Because here polymerase, that DNA dependent DNA polymerase enzyme, polymerization starts from this side to this side. When it opens to this side, a small fragment is formed from this to this side. Then it opens this part is open, then a small fragment is formed. Here it discontinuous means small fragments are formed. So this look here, this one is continuous strand. So continuous strand is called leading strand. And this side what? These are discontinuous strand. So this continuous strand, they are called as lagging strand. Lagging strand, right? And these fragments, 1, 2, 3, these fragments are called Okazaki. Okazaki, Okazaki fragments, right? And finally, ultimately what happens? These are bind with each other through the enzyme. Which enzyme is here? Here, ligase enzyme is used, because here DNA fragments are attached with one another. So this ligase enzyme is called DNA ligase enzyme. Okay. Now come back to, because here in NCERT it is given very less information. So I want to tell you about the detail about DNA replication. Okay. So see this one, how we DNA replicate itself. Okay. Now we start here DNA replication. The first process which happens here, it is unzipping process. Here how we DNA replicate? The first process, following steps, unzipping of DNA. Unzipping means what? A DNA strand, it get open. Right? Here you understand, here this DNA, this is the DNA. This one is called replication fork. Right? Because here hydrogen bonds are break down, then a DNA, that means this DNA is open. This one is called unzipping. Right? Like a zip. Unzipping a DNA, who is responsible here? Here, for unzipping a DNA, with the help of which enzyme? Helicase. This helicase enzyme is responsible for the unzipping process. It breaks down the DNA, right? Second here, this helicase, because it is responsible for unzipping, so this enzyme is also, sometimes it is also called as sweevelase enzyme, because it opens the DNA helix, right? First, not only this enzyme is responsible, means this enzyme is responsible for unzipping only. When it is unzipped, then what happens? A tension is formed. Tension due to the supercoiling structure of DNA, right? So this supercoiling structure, it forms a tension, and this tension is released with the another enzyme, and that enzyme is called as topoisomerase. Second one here, which enzyme here? Topoisomerase, right? In eukaryotic cells, this topoisomerase, right, it release the tension created by which supercoiling, by supercoiling of DNA nucleosome or chromatin, we can say here. Chromatin structure means what? It is supercoiling of DNA fragment, right? The topoisomerase is there, second enzyme, right? In a prokaryotic cell, this topoisomerase, it is in eukaryotic cell, but in prokaryotic cell, this topoisomerase, it is called as DNA gyrase. Right? So DNA gyrase enzyme in case of prokaryotic cell and topoisomerase in case of eukaryotic cell, they release the tension due to the supercoiling of DNA chromatids. That enzyme is here also. It is called SSB. SSB means what? Single strand binding DNA. This is called single strand DNA binding protein. Full form is single strand DNA binding protein. Right? What is the function of this one? When this fork is formed, then what happens? Because they are hydrogen bond, when they get free, so they have a tendency to come and forms once more this hydrogen bond. But this SSB or single strand DNA binding proteins, it prevents the formation of the reformation of hydrogen bond between two strands. Right? SSB, this enzyme is also responsible. Helicase, topoisomerase in case of eukaryotic, or DNA gyrase in case of prokaryotic. Another one is our SSB, right? These are enzymes. Okay. Now you understand here, other enzymes are also responsible for the unzipping of DNA. They have some associate works. Okay, this is the first unzipping of the DNA. Who is responsible here? Helicase enzymes. With the help of helicase enzymes, these hydrogen bonds are broken down, right? And the tension which is obtained, which comes due to the supercoiling of chromatin threads, it is released due to the presence of topoisomerase in eukaryotic cell and due to the DNA gyrase in case of prokaryotic cell. Third thing is also there, this hydrogen bond formation of, that is, we can say reformation, because one time it is breakdown. The reformation of hydrogen bond, it prevented by the another enzyme, or we can say proteins, that is called SSB or single strand DNA binding enzymes or proteins. These are proteins, specific proteins are there. This is the first step, that is unzipping of DNA. Now second step. Second step means what? It is in formation of a new chain. Second means initiation, we can say, or formation. Directly come to the formation of a new chain. Right? In the formation of new chain, because DNA, here what happens? DNA needs, what DNA needs? It needs specific RNA, segment of RNA to initiate the process. Replication process. So what happens? First, it forms DNA, forms RNA primer, right? With the help of help of RNA polymerase. RNA polymerase, and this RNA polymerase is also called as sometimes primase because primer is formed, right? Then this RNA primer, it gets separate, right? Third, what happens? This RNA primer gets separate from DNA, right? And what happens? Replication starts. Because this RNA, it also needs the triphosphate, right? So this first, it starts the process. Then what happens? Then after this DNA-dependent DNA polymerase enzyme, it continues the process. So this is only responsible for this one. This is only responsible for initiation. This one is only responsible for initiation. Then there is no work of this, right? Sometimes DNA primer is also used, but DNA primer is used in biotechnology. Naturally, RNA primer is used, right? Biotechnology means that time what happens? That time more energy is also released. At that time we use thermus aquaticus, right? We will read in another chapter, that is biotechnology. Okay, here you understand RNA primer. What is the function of RNA primer? It is responsive for the initiation of DNA replication. Now what happens? Triphosphate. What happens? Nucleoside triphosphate. This nucleoside triphosphates, right? That means how many nucleoside triphosphate we need here? ATP, we can say here deoxy ATP. Second one is our deoxy GTP. Third is deoxy CTP, and fourth one is deoxy TTP. For the formation of DNA, we need these four nucleoside triphosphate: deoxyadenosine triphosphate, deoxyguanosine triphosphate, deoxycytidine triphosphate, and deoxythymidine triphosphate. Right? Obtained from, they are obtained from nucleoplasm. So they are present inside the nucleus, right? Nucleoplasm. Then what happens here? These triphosphates, because they have a double purpose, they have two functions. Double purpose. What is the double purpose of that one? Is our nucleoside, dinucleoside, DNTP. You know DNTP means what? Dinucleoside triphosphate. What is the dinucleotide triphosphate? Double function is first and second. First function, act as substrate, and also second function is they provide energy. This energy is released when their two terminal phosphate groups are removed, right? When these two terminal phosphate groups are removed, then what happens? It becomes TMP2, like DATP2. We can say here that is deoxynucleoside triphosphate. Now it convert into deoxynucleoside diphosphate. Now it convert into deoxynucleoside monophosphate. Right? Here what happens? One inorganic phosphate is removed. Here what happens? One inorganic phosphate is removed. Right? It is also enzymatic activity, and here the enzyme which is required for this process, right? For releasing of these two phosphate group, which enzyme is required here? This enzyme name is pyrophosphatase. Which enzyme? It is pyrophosphatase enzyme. Due to the presence of this pyrophosphatase enzyme, these triphosphates, nucleoside triphosphate, they convert into nucleoside monophosphate, and the release energy, a huge amount of energy is released, because this DNA replication is a very expensive process when we consider the energy. So this energy is provided through nucleoside triphosphates, right? This energy. That means what? You understand here, DNA replication, for the formation of, for the process of DNA replication, DNA is not dependent on mitochondria for ATP, for energy, right? So DNA need not mitochondria's ATP, right? It is totally independent. It get energy when the triphosphates are converted into monophosphates, right? Now a long chain is formed. Now second, what is happen here? Now another process is here. How a elongation means this, because here DNA is double stranded structure. Two strands here. I am going to draw here, and you understand. Look, this is the DNA. This is the DNA replication fork. These two strands are get separated. This side, this one the mother DNA. Right? Suppose that I write here adenine, guanine, cytosine, thymine, adenine, cytosine, guanine, and so on. Adenine, right? That means what? Here what happens? Just complementary, because complementarity is there. We know very well. Complementary. So which one is complement here? A. Here it is T, then C, then G, then A, then this one is T, then G, then C, and then T. This is the sequence, complementary sequence. A is always bound with T and C is always bound with G. We know very well. Right? Now the daughter strand is formed. When a daughter strand is formed, so that is this one. So first, triphosphates, they convert into monophosphate, and this monophosphate is attached with these nitrogen base, forms a pair due to their complementarity. That means A always attached with TMP, trimonophosphate, right? Deoxytri, that is where thymidine monophosphate, deoxythymidine monophosphate. This side what? It is adenosine monophosphate, deoxyadenosine monophosphate. This is the G. G means what here? Deoxycytidine monophosphate. Here deoxy G guano, that is guanosine monophosphate. That side T is there. It binds with deoxyguanosine monophosphate. It binds with deoxycytidine monophosphate. So in this way, monophosphate, these are attached with them, but they are not forms the bond in between them. After some time, what happens? A bond is formed, phosphoester bond is formed in between these bases, right? When a bond is formed, now this becomes strand. First thing, okay, suppose that this one is our 3' and 5', and this side I write here 5' because they are antipolar. So another one is, and this one is 3'. Here one thing is there, DNA dependent DNA polymerase, DNA dependent DNA polymerase enzyme. It acts only in one, that is 5' to 3' direction, right? So 5', this side is 5' and this side is 3'. So what happens here? Here starts from this side, not from this side, but from this side. So here what happens? Here a continuous strand is formed like this, right? We can write here because it is D, then it is A, T, G, C, T, right? And this side what happens? This is the continuous strand is formed. Now this one is it opens more and more monophosphates are formed, right? But in this case, what happens? Here, what happens? Here, because DNA dependent polymerase enzyme, it acts only in 5-3 direction, means it starts from that side. So look here, when these three nucleotides are open, a strand is formed, a small strand is formed, right? Then after, what happens? When these are open, a small strand is formed. Now then, this small strand is formed. What happens here? Here a small fragments are formed. These are the fragments, DNA fragments. This one is called discontinuous strand. Discontinuous strand, right? So this one is, another name is here lagging strand is formed, and this one is continuous strand. This strand is called leading strand. Leading strand, right? Because here fragments are formed. So these fragments, these fragments, they are called as Okazaki fragments. So with the help of ligase enzyme, which enzyme? With the help of ligase enzyme, ultimately they form continuous chain with the help of ligase enzyme. Which enzyme? DNA ligase enzyme, right? These fragments, they join with each other and form a continuous system. So here, when you see, you can understand, because this one is A, T, this is A, T, this is G, T, it is A, right? G, C, C, G, T, A. A strand is formed. When you see this one, so that means what? T, T, A, T, T, A, C, G, A, C, G, A. On both strands, A, A, T, G, C, T, G, C, T, means two identical strands are formed in both strands, right? In DNA replication, and this strand is called, this one is parent DNA, parent strand, or we can say mother strand, right? And this strand, it is called as daughter strand. Right? Because in new DNA, when these two DNA, they get separate, right? So this in new DNA, one is parent, another one is offspring, or we can say one is mother strand, another one is daughter strand, right? Only one strand is formed, another strand comes from the mother strand. That means what? Here we can say it is a conservative. This one is semi-conservative replication, right? So this one is called as semi-conservative DNA replication. So do you understand how a DNA replicates? I think now you have no problem. Okay. One more thing which we need to discuss here. In DNA replication, we have three types of enzymes. In case of prokaryotic cells, so DNA polymerase enzyme is three types, right? So three types of DNA polymerase enzyme is what? DNA polymerase first, DNA polymerase second, and DNA polymerase third. DNA polymerase first is responsible for initiation. DNA second, it is the least activated. It only activates when DNA polymerase first and DNA polymerase second is absent. So in the absence of polymerase first, polymerase second, this DNA polymerase second is activated. And this the DNA polymerase third, it is the main enzyme which is responsible for the elongation of the chain, right? Here, another point is, there are three types of DNA polymerase in prokaryotic cell. I write here in E. coli. Simply E. coli means to understand it is for prokaryotic cells, right? Which one? First is our DNA dependent. Here I write only DNA polymerase, right? So this is DNA dependent DNA polymerase. So DNA polymerase first, second one is DNA polymerase second, third one is DNA polymerase third. Three enzymes are there, right? So this first, it is responsible for initiation of replication. This one is only active in the absence of polymerase first and polymerase second, right? DNA polymerase first, so it is activated only when polymerase first and polymerase second is absent, and DNA polymerase third, it is the chief or main, it is the main enzyme. For elongation of DNA, right? You understand here? These are the three types of DNA polymerase enzyme. This DNA polymerase, because it is discovered by Korenberg, so it is also called as Korenberg enzyme. Korenberg enzyme, because it is discovered by Korenberg, right? So Korenberg enzyme is also called as Korenberg enzyme. Okay, so this is the DNA replication, and I think you understand all about the DNA replication.